This is TWIS, This Week in Science, episode number 731, recorded on Wednesday, July 24th, 2019. Archaeology from space. Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with parenting, females, and a locker. But first, TWIS is supported by listeners like you. We thank you for your support. We really couldn't do it without you.
Disclaimer, disclaimer, disclaimer. The following acts of communication are all in your head. Nothing you are hearing now is happening now. Not even the sound of this voice saying these words. The person most often associated with the sound of this voice is not actually saying this right now. In fact, his lips may not even be moving. What you are listening to now is a recording of a voice that in a short while will be accompanied by other voices, all telling you about the amazing things that are happening now, or happened long ago, or might happen in the future. As we speak, these voices are taking up residence in your head, re-wallpapering the interior of your skull, setting up subtle sciencey subroutines in your subconscious mind, navigating your every third thought in the direction of This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. It's time again for another episode of This Week in Science. Oh, we're back again after another week. So much fun. So much fun. So much science. I brought stories about fake visions and parent brain. And we have an interview this evening to discuss space archaeology with Dr. Sarah Parcak. That'll be coming up in just a few moments. Justin, what did you bring?
I didn't know space archaeology was the thing I could have done. That sounds really awesome. I can't wait. This is a synthetic protein that can work inside living cells called LOCKR. That's going to be really exciting. There's a study looking into global warming. Spoiler. It confirms it, but gives some explanation for some of the decade-long shifts that we've seen over the last few thousand years, and something from the Scientific Journal of Marketing, of all things.
Scientific Journal of Marketing. You know, marketing can be very scientific, Justin, so I'm looking forward to hearing all about that one. Blair, what's in the animal corner?
I have sex-changing fish. I have screaming embryos, and I have some climate change.
Oh.
Mm-hmm. Climate. I don't want any more climate change. Can we?
Well, I have climate change news. I'm not bringing you more climate change, just some science related to the inevitable effects.
We have enough of the climate change coming without any more. But yes, okay, news for that. I will look forward to it, as well as the other Animal Corner news. As we jump into the show, I want to remind everyone that if you have not done so previously, you can subscribe to This Week in Science. All places that podcasts are found: iTunes, Google, Spotify, Spreaker, radio.com, Pandora, TuneIn. Just look for This Week in Science or TWIS. Also, you can go to YouTube and find us there. We're also on Facebook. You can find us easily at twis.org for information. Okay, now it is time for our interview. I'd love to introduce our guest tonight, Dr. Sarah Parcak. Dr. Parcak is a space archaeologist, and like Justin said, maybe many of you didn't know this was a thing that you could do. So we'll talk about that. She's a professor of anthropology and director of the Laboratory for Global Observation at the University of Alabama in Birmingham, and expert in using satellites for remote sensing and in archaeology and Egyptology. She's a National Geographic Explorer and a TED Senior Fellow. won the 2016 $1 million TED Prize, which that money was then used to launch a citizen science platform called Global Explorer that aims to bring the wonders of archaeological discovery to everyone. And she has a book that's out now called Archaeology from Space: How the Future Shapes Our Past. Dr. Parcak, thank you for joining us tonight on the show.
Thank you so much. And can I just say I'm renaming my garage band name to Screaming Embryos?
Yeah, I know. That's such a great.
That's a good one.
Yeah. You're welcome.
Oh, for all the good band names, come to This Week in Science.
Yeah.
Just go to Blair. Just go straight to Blair.
I have a
She has
list.
them all.
All right, to get started, I'd love to know first, what is a space archaeologist and how did you become one?
So it's kind of the super fun public facing name of what I do. Like most scientists, I wear a lot of hats. I'm an Egyptologist. I'm a landscape archaeologist. But my specialization is that I look at different types of satellite imagery to try to find subtle hints of entire ancient settlements or buried features on sites, or even environmental features like rivers that may indicate where humans used to live. I did not come up with the term space archaeology. That is NASA's fault. They actually, yes, it is their fault. They have a space archaeology program that funds exactly what I do. And if NASA calls what I do space archaeology, ergo I am a space archaeologist. Look, yes, technically I'm a mid-troposphere, multi to hyperspectral pixel surface feature detection analyst, and now all your listeners are asleep. Space archaeology, let's get all the kids excited about science. That's kind of whatever it takes.
Absolutely. How did you get interested in doing archaeology in this particular manner?
The reason I do what I do is because of my grandfather. He was a forestry professor at the University of Maine in Orono and a member of the 101st Screaming Eagles in World War II. And he pioneered the use of aerial photographs and the use of analyzing old forests. So he used overlapping aerial photographs to measure tree heights. And he's the reason I took my first remote sensing class as an undergraduate. So at the time, high resolution imagery was super expensive. I mainly looked at NASA satellite imagery. So it was really, really just getting going in the early 2000s.
So it could have been quite called earlier aerial archaeology.
That's right. So, and that's still part of the field of archaeology, aerial archaeology, the field of interpreting aerial photographs. Yeah. Obviously, very important even now, especially as we look at old photographs from the sort of 50s and 60s that indeed show landscapes that no longer exist. They're helping archaeologists map sites. In fact, one of the coolest satellite data sets I use is high resolution black and white corona spy photographs from the 60s and 70s. And I get to look at old images of Egypt and map parts of sites that are largely gone.
Right. So there's places that are parking lots or housing developments and all this stuff. And then we've sort of lost any trace of what could have been there. And by analyzing these old images, it becomes maybe apparent to somebody with a trained eye.
That's right. And what we're looking for from space, first of all, we're looking for the chemical signatures of entire sites. So everything on the Earth's surface has its own distinct chemical signature. And I'm using the same images, processing techniques, computer programs that, say, biologists use when they're looking at vectors for animals or diseases, or urban scientists who are looking at change detection, people who are looking at mapping the polar ice caps over time. And when I say chemical signature, in the light spectrum, different things spike in different parts, whether it's the near, middle, or far infrared. And sometimes entire archaeological sites will have their own distinct chemical signature. Sometimes things that are buried under the ground will be reflected in different parts of the light spectrum. So think of it kind of like a cool space-based CAT scan, where things buried under the ground will affect the overlaying vegetation or soils or whatever, or geology in ways that we can't see with our naked human eye, but we can in different parts of the light spectrum that we can see in different kinds of satellite imagery.
And one of the examples of this that jumped to my mind was the longhouses in Newfoundland. Where they identified what looked like the structures of Viking type settlements on islands that they hadn't ever considered Vikings to have gotten to, but the outlines that they could see somehow from the differentiation of the soil kind of painted the picture of a typical Norwegian or whatever Viking longhouse settlement. And then, of course, people have to go into the field, get on boats, go out to these islands and do some digging, but actually found evidence that these stones were placed here, and this is, they had foundations and the like.
So in archaeology, no matter where we're looking for anything, whether it's in Egypt or Newfoundland or Peru or China, it doesn't matter. We're going from the known to the unknown. So before we even start this project work, obviously this is science. We don't necessarily go somewhere because, gee, there's something cool there. We go because, we're interested in answering a question. We develop a hypothesis about that place. Maybe this settlement moved here X period of time. Maybe we think the civilization collapsed for different reasons, and we're trying to find evidence for that. So we develop databases of dozens to hundreds of known examples of different sites and features. And so before we even begin processing, we have to think, what are we finding? And then we look at the area's geology. We look at weather patterns. We look at the soil. We look at seasonality. That's a big deal. You might find something in spring that we may or may not find in the fall. And that's before we even order the imagery. So it's a really detailed, almost kind of like a detective process that we work through before we actually start analyzing the imagery.
I'd love to know more about Global Explorer, though. So from the science perspective, it's hypothesis driven. You're following a trail of people in a particular area looking for evidence of their activities. Is it the same kind of idea with the citizen science projects that you've got on Global Explorer, or are you really just kind of using people's eyes to get you images that might be useful later?
So when we were developing Global Explorer, we wanted to create an experience for the people who would be using it that would be very similar to a remote sensing scientist. Now, it's not like they're getting on the platform and they're doing, advanced algorithms to find a specific tiny feature on a single site. What they're doing is they're looking at the same level of zoom that we would do for a satellite image. So that's about the right distance if you get on. And then they're determining whether or not there are specific shapes or features in that satellite image. So this is very high resolution, so 0.5, 0.4, even 0.3 meter resolution data. Is there something there that merits further analysis? And what we're doing is, it's not just like one person can see a thing and then it gets kicked to us. A minimum of 6 people need to look at a single satellite image, and 5 out of the 6 of them need to agree that it either is a thing or isn't a thing. That's just the number that we came to over time and talking to other sort of online citizen science projects and PIs. And in a way that sort of gets around, in some cases, the lack of expertise. If 5 out of 6 people say it's a thing, what we found when we looked at the back end, when we get the data, that our users have between an 85 and 90% success rate in terms of detecting features. So they're really, really good at it. And obviously you get better over time. And so what we're doing is we got areas of interest from the Peruvian government, from the Ministry of Culture. So we took that information or that desire for more information from them. We're working very hard to collaborate, of course, with ministries wherever we go. And then we set the eyes of our crowd on those specific places. So then what we did is we shared the data with archaeologists on the ground. And in one case, working with this amazing gentleman, Dr. Luis Jaime Castillo, who's now the Minister of Culture in Peru, he used the information from our crowd and did additional drone mapping that led to the discovery of 50 new Nazca lines. So this idea of collaboration, empowering local archaeologists with the data the crowd gathers. So there's sort of like we've had to learn a lot about citizen science as we've gone along. The ethics around it are murky. There's a lot of give and take. How do you respond to the crowd? How do you give them and thank them and make them feel like they're part of this larger scientific effort, which they are. And the goal once we kind of sift through more data is to do a huge peer-reviewed publication, and all the citizen scientists should be authors, in my opinion.
I think that's wonderful. There have been a few publications that have come out of these kinds of citizen science endeavors that have published with the long list of names of citizen scientists who have been involved, and it's pretty neat to see. For your project that you have up now, I was digging around on your Global Explorer site, and it's currently a question of looking for looting versus not looting. Can you talk about the problem of looting and what you're hoping to find with this particular project?
So on the platform, one of the things that we grappled with and are still grappling with are user engagement and user experience, so UX and UI. And what we decided to do was it's not like people are just going on the platform and they're clicking buttons and that's it. It's actually a game. So there are 10 distinct levels. The more tiles you examine, the more you level up. And so you start off on the looting level, and you're just looking at this image. Is there looting? Isn't there looting? And the thing about looting is it looks the same everywhere in the world. So it's holes in the ground surrounded by a donut shape of earth. I should say, too, for the people that are listening, it's not just like we throw you on the platform and start. You take several tutorial videos to show you different examples of sites and features in looting. And unfortunately, looting is a problem all over the world. Certainly, it's a huge problem in Peru. We see a lot of objects on the black market here in the US and in other places. And so by determining that there's looting going on at a site, that information can then be shared with government officials. It's not up to us to prosecute or investigate. That's always up to the governments. But at least, we have a good map of where it's going on. And if you have a map of where it's happening, then you can take steps to hopefully do something about it. So after I think it's 1,000 tiles, then you level up, which is when, of course, you can start looking for sites.
But is the problem of looting, is it a really bad problem? I mean, we've got movies like Tomb Raider, and even with Indiana Jones,
Andy had
there
some
was there was looting of
belongs in a museum.
Problematic. I will admit that. Totally problematic, Dave. I mean, so looting is not a black or white issue. There are many, many shades of gray. The first people that most people in the West tend to blame are the looters themselves. I don't blame them. I blame the Western collectors. If there's no market, people aren't going to loot. And then there's this picture painted of, poor, desperate local people who are doing it to survive. That's also not true. Looting is in many cases like a gig economy. You need to get some money to maybe send your kids to school or an operation or something short term or quick. I think we in the West have a lot to answer for. Look at Christie's and Sotheby's. They're always in the news because they're yet another object has had a falsified what we call provenance or area of origin, and it was very likely looted. So what I do whenever I speak publicly is I say to people, why would you want to own a piece of the past? Basically, you're Gollum. And you want to sit in your lair and stroke the ashes. Like you take it out and you show your rich friends, and yay, you have a thing from Palmyra that ISIL looted, and congratulations, you funded terrorism. Versus, why don't you take that $1 million or whatever you paid for it and fund 20 excavations for multiple years of digging, and you at your fancy people rich parties get to brag that you helped to rewrite history.
This is very similar to how we talk about poaching, right? Yeah. Why would you want to own that dead endangered animal in a rug or in a taxidermied item instead of using that money to preserve that species? And then I don't know if you really want to take your buddies on a safari to go see them, go ahead, but that does way more good, right? Yeah, it's absolutely it's very similar.
So the juxtapose here I'm hearing is that science just isn't paying enough. Because the early days of archaeology, then it kind of is sounding like, I mean, how did so many Egyptian artifacts end up in British museums?
The British Museum.
It was technically the version of looting, I guess. But the preservation aspect and the public aspect and the transparency thing, I think, would be the differentiation when we're talking about this.
Right. And also, like, rampant colonialism. I mean, obviously still a huge problem. We're a lot of us are fighting to decolonize the field. But this ironically, like, in spite of all the issues, challenges, problems, this model that existed through the 30s and 40s and still does to some extent today of very wealthy donors and sponsors helping to support archaeological research. I think that's the wave of the future, whether it's microfunding through Kickstarter or GoFundMe or whatever it is, or donors. And fine, they can come to your site. They can come clean pottery for a day. There just isn't the government funding anymore. It's drying up. So we need to think about better models for sustainable funding to help support this work because so many scientists are struggling right now.
Yeah, and you're also dealing with a world that is rapidly changing. We have climate change where many areas that might be of interest will be underwater soon, and what you'd have to be a marine archaeologist to be able to reach them. We've also got war. Like you mentioned Palmyra, which is also something you talk about in your book, that it is the idea that things are being blown up and things are being destroyed by people. Is there a feeling of time pressure, of that, you don't have the money, but you also want to try and record things and see what you can find before it's too late?
Yeah, I think, not to be dire, but we who are in the science sphere know we are running out of time. The oceans are going to rise. There's a glacier the size of Florida that is breaking off right now. And we are seeing levels of heat in the Arctic that were not expected for another 70 years. Mm-hmm. I think we need to decide what we're going to save and how we're going to save it now, because we can. There's time. Looting and climate change are two issues, but right in the middle and the much bigger problem is development. Mm-hmm. Urbanization, sites being bulldozed through deforestation, it's all kind of tied together. And the biggest problem from what I see is if you don't have a good map of where your cultural resources are, how can you know what to protect? So most countries in the world don't have good archaeological maps, which is pretty shocking. Okay, so we know in general, if you ask a biologist, how many species of X are there? And they would be able to tell you down to the specific bird type or slug type. How many archaeological sites are in the world? I don't know. I'm an archaeologist. My job is to map thousands of sites. I have mapped sites all over the world. I have a rough guesstimate in the book, and I have a rough guesstimate for how many sites I think there are left to find. So anyway, that's what I think the issue is.
So I have a proposal that may horrify you. But this is where how my brain works. If we identified sites most at risk, could we partner looters with scientists archaeologists who could do some preliminary verification, identification, and therefore themselves get to do some analysis and register that this existed at one point, this was found, which would then increase the value of the looted object because it would have a verification behind it, but do so in the most at-risk places in order to loot them as quickly as possible, because government funding and the like is not going to be sufficient to actually excavate those sites now, with the hopes of at some point later in the future, being able to recover those objects if even necessary, but having at least gotten a chance to record them. Partnering with looters, I
can do better than partnering with looters. Why don't we create a global citizen archaeology movement whereby in communities on or around archaeological sites, we empower the citizens there with the tools and resources on a phone. Just create an app. All they're doing is they're the cultural stakeholders. They're going to sites. They're documenting, recording. I'm going to say a word that will make you all twinge. Blockchain. Most uses of blockchain are complete baloney. I don't know what words I'm allowed or not to say on the show, but I'm going to use the word baloney.
That's a fine word.
but so when you put something on chain, it's immutable. You're recording the exact time and place of that thing. And one of the really cool uses of blockchain, I've become convinced or converted. What if every archaeological object and site in the world were on chain? What if every part of every monument of the world were on chain? That way there's a global immutable database. So blockchain, if you imagine like a global antiquities registry, and you have all the objects everywhere in the world that are on chain, and these citizen archaeologists have helped document them everywhere because you've given them training and they're going out. The darn archaeologists in the world to help map all these sites. So we have to train other people, and archaeologists need to they got to get out of the ivory tower. We've got a decade, maybe two, to document as much of this stuff as possible. And why not create millions of people around the world that are really excited about archaeological sites and they're taking part in documenting them? Like, why isn't that a good thing? These people are not interested in the third regnal year of Amenhotep III. They're just not, generally. I might be, but they're not. So let's get out of the ivory tower and get this out of our heads. So
do you think that will it'll get you out there doing stuff? Do you think it's going to reduce the quality of the science that can be done? Or do you think you just need to get a higher throughput?
I think we need a higher throughput, in so many places. We just need people documenting the sites, their condition, what's at risk. With so many limited resources around the world, archaeologists more often than not, they may work at one site, but there's another site that's far more at risk. And if they're working there, at least they can take the steps they need to protect it. So like I said, it all starts with a good map. And, for example, we're going to India next with Global Explorer. We're working very closely with the Ministry of Culture, with the TWIS, which is an enormous not-for-profit organization. They're providing very generous funding for what we're doing. And the Archaeological Survey of India, the body that is in charge of of India's heritage, has asked us specifically to help them create a national database of sites. So again, it's coming from them. We're not going in with like a lot of Westerners have done in the past. So it's something that we can do. It's a resource we can provide to them so they can do a needs assessment for what's there. I have this crazy dream of mapping the world in the next 10 years. We're doing machine learning, I should say, as well, to go through more data that'll be built into the back end of the platform. We're rebuilding it right now so that we'll be able to go through things even faster. We're doing everything we can to make this more efficient. But yeah, in terms of the science, the science will happen. I just feel like, I don't have to put the science on hold, but yeah, I feel like we're in a crisis right now.
Yeah, I'm just thinking there's Blair brought up the similarity to conservation biology and poaching, but we also in biology there are experiments done where there's just data collection, genetics, genomics. Sometimes just collect all the genetic data and then do the experiments. And so maybe that's where archaeology needs to be for a little while to get all that information out. In your book, you talk about the idea of how the future shapes our past, but technology has been such an important part of what you're able to do and what you are going to be able to do. Like you just mentioned, machine learning. How do you see technology influencing your field in the years to come?
I actually have a whole chapter on what archaeology is going to be like in 100 years. I'm a total sci-fi nerd. Like, I grew up on Clark and L'Engle and so many others. And today I read Jemison and Stevenson. And anyway, I've always wanted to write science fiction, and gosh darn it, it was my book. I was going to write science fiction. So I invented all these things that we're using: satellite imagery, DNA analysis, chemical testing, radiocarbon dating, even things like reading burnt scrolls using CAT scans. I imagine this guy named Robbie who works for a company called Archaeotech, and he's rapidly exploring sites, and he's got these things that I call dig bots, which I should totally trademark. He goes to these sites and he sends off these little mini drones, and they're mapping the sites quickly, and they're measuring everything that's underneath, and he's sending up dig bots to go rapidly excavate. And in an hour, he's able to get as much information as we archaeologists would get in 100 years at a site. But then what I do in the chapter is I kind of stop. I'm like, well, y'all think I'm probably like on something, but here's where archaeology is now. Here's where the technology is. Here's how quickly it's advancing. And I think we'll get there faster than 20 years. And I think we archaeologists are not going to be exploring sites on Earth for the purpose of exploring sites on Earth like we do now. In 100 years, I think we'll be preparing to look for civilizations on other worlds, assuming we get through the climate crisis. And NASA doesn't have any archaeologists on staff right now, which is an enormous problem. We're the only ones on this planet that have the framework in place to contend with any civilization at any stage about which we know nothing, which is what we're going to find in other worlds if it's beyond multicellular life.
Right. If we get our telescopes working, or even get our craft out to rendezvous with other places, these exoplanets that we're looking at. That, I mean, I know from the biological standpoint, or the chemistry of life standpoint, we can look for the spectra, the life signatures. Right now, we're looking for methane, we're looking for water in the atmosphere, but your ideas would go beyond that.
I mean, yeah, and again, it's total speculation, because like, I don't eat octopus anymore, because I'm like, oh my God, I've been eating like the smartest animal that actually apartments for themselves under the ocean. I'm like a cannibal. So yeah, we have no idea what these settlements will look like if we happen to get that sweet spot of when that civilization exists. But, put us archaeologists to work. We have really good imaginations. It's worth a try, right? I mean, we have no idea what shapes or sizes the civilizations or features will be. But I'd like to think that we know a thing or two about creating something from nothing.
I think so. In your research, I mentioned Lara Croft and Tomb Raider and Indiana Jones. Lara Croft, I remember when I was younger, she was a very, I thought, even though it was a video game character extraordinaire, I was excited about seeing a woman in a main character archaeologist's role. I mean, she was raiding tombs, but still, I was excited to see. I was excited to see a female in this kind of role, whereas Indiana Jones and others, and even historically, archaeology had been very male dominated. Do you see the field, like you mentioned, colonialization? Do you see a change in the representation of the sexes in archaeology as well?
So in most anthropology, archaeology grad programs, I can only speak to universities that are, say, in the UK or in the US, although I have to say my colleagues in Europe generally, I think, would speak to the same trends. 70+% women or more, which is great. But then as they advance, there's massive attrition rates, due to abuse, lack of opportunities, sexism, all the issues and challenges that have kept women out of the sciences or prevented them from entering and then of course, even if women are get academic positions, or is there an extra year added on the tenure clock so they can have a kid if that is what they so choose? And. So I am a rare beast, and I don't know what percent of women who do archaeology end up as professors. I should probably figure that out, but it's a very small percentage. So I do my best to speak up about the sexism. I do my best to, try to encourage and mentor as many junior scholars, especially women. And I make a lot of middle-aged old men in my field really uncomfortable because they're fault, and I call them out on it all the time. So it's all, you pave the way and you try to make it better and easier for others. Because
yeah,
it's kind of interesting. I think that by my list of archaeologists and anthropologists that I know and have met over the years, I would have to say it is in the 90% female category. I don't know if that's a California thing, but it's I somehow have this picture of this being a female dominated field, and it could just be completely my own personal perspective of what I've encountered.
Or it could be the psychology of you seeing 50% women and your brain tells you it's more. Which is the thing.
No. But okay. Also, having had been in a relationship for a long time with an archaeologist, anthropologist who was female, who had female friends, that's probably the network. That's probably how that happened.
Probably how that happened there.
But even the lead on the research that was the professor at UC Davis was female. So this is. Yeah. Anyway,
I think you're talking outliers for sure. Yeah, but I do think there's this larger question of exactly, Sarah, what you were getting to, where in academia you tell women that they have to pick between taking advantage of their baby making window for their body or continuing in academia. And I think there has to be a solution to this problem. I look forward to hopefully being able to future generations, this not being so much of an issue, but it is something that I know several people who've been pushed out of that path because they decided to take advantage of their biological window to start a family.
I have a super controversial thing to say about this.
Okay.
Great.
I benefited from the solution that I'm about to propose. So I got my PhD at Cambridge, and between my MPhil and my PhD, I was only there 4 years. It did not take me 7 to 8. I think we waste time in grad school. Mm-hmm. I finished my PhD and I got an academic job. I was really lucky. I know it's a crapshoot, but like, what would 3 to 4 more years of grad school have done for me? Taking a few courses. I know how to look things up. I know how to figure out problems for myself. I think we need to lessen the time in grad school across the board. The reason that I think I was able to choose to have a child was because I got tenure around the time that most people are starting academic jobs. And I was a super freak genius. It's because I finished my dissertation when I was 26, started a job when I was 27, and had tenure by 32, 33.
Yeah. Yeah, that makes a big difference. It absolutely does.
I'm going to throw out something even more controversial to end it. Right? Something even more controversial.
Justin will not be one-upped in this arena.
I won't be one-upped by controversy. The other thing that can make a woman very successful in having children and maintaining an academic life is having a good man.
Or partner. A good partner.
Partner.
A good partner.
Sure. Yes. But I schlepped my 2-year-old son across Greenland while his mother was doing field work. And this is one of those things that if she didn't have a guy who could leave his job behind to go schlep across Greenland with a 2-year-old.
Justin. Pat yourself on the back there. Good job. But yes,
it's behind every strong anything is somebody who's helping them, be that. And it's a partnership is like you're saying is crucial in any of those scenarios.
Right. No, look, I have an amazing partner. He's more of a traditional archaeologist. So in other words, he doesn't need the fancy computer equipment and programs that I do. He's more of a traditional scholar. I tell people that his articles are things that we've footnotes together. Like he's insane scholar. But he's a wonderful partner and incredibly supportive, and I couldn't do what I do without him. It's hard for women that have 9-to-5 jobs.
Yeah, it's hard. It's hard for everyone. There's to find that balance.
Yeah.
Here's the thing. Here's a word I am. If I could get one thing, get on the soapbox tonight. There is no such thing as balance. Balance assumes that life stops. It's a pendulum, and sometimes it'll go like this, and sometimes it'll go like this. But you don't want it to go haywire. You just want to predict the pendulum. Ooh. So, a little nugget of wisdom that I will share with you tonight.
Nice.
I appreciate that. I'm always looking for good
remember that one.
I know. Come on, Kiki, stop looking for balance. Just swing with it.
Yeah. It's 7:30 p.m. and I'm still at work. The pendulum's just swinging this way tonight. It's all right. I'll fix it tomorrow.
I always thought balance was making a successful blanket fort in the living room.
Oh, yeah.
And hiding there for the evening. That to me was balance.
Still fun. I'm still with you on that one.
Yeah. That's how you find balance. Hide in a tent you made in your living room.
And then somebody will look for the archaeological remains of it at some later point. Is there anything else you would like to tell us about the book that you've written and what you hope to get across in the book to people who read it? I
hope that the book will show people what archaeology is really like. It's about this new science, but I talk a ton about what digs are like, the types of people you find in excavations, how we archaeologists take all this information and piece it together to try to recreate stories of what happened. I want people to see the richness and the vibrancy of archaeology and why aliens didn't build the pyramids, people did. I want, the stories that I tell about the past, and I have there's not just a science fiction chapter, there's a fiction chapter where I bring to life a 4,200-year-old anonymous woman from the Egyptian Delta. I hope that by learning her story and the story of her family and her struggles in a period of great climate change and upheaval, and then kind of connecting that to the archaeological information, I hope that creates some empathy, right? Because I think the world now needs a lot more empathy. We so many people who think it's okay that people belong in cages. Their empathiometers are down to 0 because they can't see beyond themselves. They don't have the ability to connect. But by understanding the past, by looking at the richness and the diversity of everything that has merged to create our very modern and interconnected, and in some ways not so modern in our world, it's because of the diversity of all these cultures. And you understand that by learning about them. And I think the other thing I hope the book does is rekindle people's ability to experience awe. I think that's missing right now in our world, in spite of us being so interconnected. You don't experience awe. It's rare to experience awe through media. I think, that the great example, of course, is the landing of the Apollo 11 on the moon, because we were all fixed. We were all experiencing this global moment in time. And the same sensation of feeling overwhelmed and feeling like you're part of something bigger than yourself, you get that when you're in front of Angkor Wat or on Easter Island or at Machu Picchu or in front of the pyramids. And I hope I inspire people to go out and experience awe for themselves, because when you feel small, it's not a bad feeling small. It means you're interconnected. It means you're part of something bigger than you ever could have imagined. And that, I think, inspires you to kind of feel like you can own your place in the world by making it better. So hefty goals for a small book, but small but mighty, as they say.
Small but mighty, and I love the so perspective changing. I mean, not just looking at the pale blue dot from space, but using our technology, our new view from space to be able to really give us a view on ourselves, who we are now, where we came from previously, the many cultures around the world and how they're interwoven in the braided stream of life, right? The braided stream of humanity. Where can people find out more about you and your book?
The book is available on Amazon, IndieBound, or ideally your local independent bookstore. Or please check out your local library. I'm a huge fan of libraries, and if they haven't ordered it, please encourage them to, or take it out. I'm not shilling the book, just want as many people to be able to enjoy it as possible. You can get it on Kindle or whatever device you use. And I am, I guess I'm highly Googleable. You could listen to my TED Talks, as they say.
Yeah.
Documentaries I've done are on YouTube. So I did one called, I'm trying to remember the names, Egypt's Lost Cities, Rome's Lost Empire, and Vikings on Earth. So they're all on the YouTubes, and a bunch of lectures and other things I've given are online. Yeah, it's slightly frightening how much information is about me out there. But I mean,
sometimes you go, wow, how did I put myself out there so much? But, I'm glad that you have, because I think that you have, by putting yourself out there as a public figure and an archaeologist, you're a role model for so many young children to look up to be inspired by, and hope that maybe one day they can be a modern day Indiana Jones looking down from satellites or using drones or machine learning. Thank you so much.
So yeah, if I could, if as a child, somebody told me I could be Indiana Jones in space, I would not have looked back. I had the hat, I had the rope swing in the backyard on the tree, and I had the theme song rolling 24/7, and vaguely understood there was some archaeology involved. But still, you put it in space, it would have been a whole different scenario.
There is a photo that I don't think I'll ever share publicly of Harrison Ford and I fighting over the photo.
Oh my gosh.
Seriously, he's a lovely man, and he did not have to take that photo with me, and he did. Nice. So yeah, and when I'm not having a good day, I'll think about that photo. I'll be like, yeah, I did that. That's pretty cool.
That's one for the pendulum for sure.
All of the good Harrison Ford roles in one.
But who knows? I think we need a new Indiana Jones movie. I think it's been a while. I think, and I don't mean like Indiana Jones in the same vein of hella problematic colonialism and small Asian children. Like the bad.
Yeah.
I'd love it for a woman of color. Wouldn't that be amazing? Dealing with looting or something. I who knows? But
who's actually done this?
Dora the Explorer.
Yeah, Dora the Explorer.
Dora the Explorer.
In a cartoon.
That's the movie coming out. Yeah,
she's got a movie coming out. But I think she's looting in the movie, which is sad. But no, maybe she's fighting looting. We have to wait and see when the movie comes out.
TWIS Movie Club.
Yeah. Rate the lootiness of Dora.
Yeah, our son isn't into Dora the Explorer, although Molly of Denali, that's amazing. My
I don't know this one.
Anthropologist goes PBS, highly recommended.
I don't know.
Yeah, that's another good new one. That's yes.
Ooh.
Yes. There are some good ones coming. I think WGBH is behind Molly from Denali. That's an awesome one. Nice. Yeah. I love it. Things are on the path to change. We just have to keep working and doing our best to help that change happen, right?
Thank you, Sarah.
Yes, thank you so much. I really appreciate getting to talk to you.
Thank you.
I would love to talk to you again sometime. I hope you have a wonderful night. We'll let you get your rest.
Thank you so much, y'all. Good night.
Thank you. Good night. That was Dr. Sarah Parcak. Her book is Archaeology from Space, How the Future Shapes Our Past. It's a really good book. I'm enjoying it a lot. It was a fun interview. It is time for us to get to some science news, however. So we're going to take a quick break. And when we return, there will be more This Week in Science. Stay tuned.
Explain things you've heard with more than intuition.
Hey, everybody. Thanks for watching TWIS or listening to TWIS. I hope that you are enjoying the show. I do hope that if you enjoy us, that you might think about supporting what we do. We do this show completely listener supported, and it would be wonderful to get your help in keeping this weekly show going month after month after month. If you would like to know how to do that, the easiest way is to head to TWIS.org, where you can find all the important links. That's right. If you're interested in buying some of our merchandise, there is a link at the top of TWIS.org to our Zazzle store, where we have t-shirts and hats and all sorts of TWIS goodness. That's right. TWISy goodies, with our logo and also with art from Blair's Animal Corner calendars. Portion of the proceeds do go to support TWIS. We also have a donate button on our main page that uses a PayPal interface, and you can donate an amount of your choosing. Also on the header bar is a link to Patreon. You can click on that Patreon link. It'll take you to our page where you can click to become a patron and choose the amount of your support. At $25 a month or more, you get one of the TWIS t-shirts. You only get charged once per month. So $25 a month, that's probably around $5 or $6 a show, which, you know, you probably spend that much on a latte on a regular basis. If you enjoy the science and the educational material that we try to bring to you, the quality we try to bring to you week after week, please consider helping out. Also, back at TWIS.org is the subscribe button. Click on the subscribe button. You'll be able to subscribe on YouTube, iTunes, or Google Play. Hey, if you're not subscribed on YouTube yet, click that YouTube link, because we are almost to 13,000 subscribers. Why don't you help us get to 13,000 subscribers? You can click and subscribe at YouTube. And also, if you do that, don't forget to click the little notification bell on the YouTube channel page so that we can make sure that you actually see our shows when we publish new ones. We thank you for your support. We really could not do this without you.
And we are back with more This Week in Science.
Yes, we are. And it's time for that part of the show that we like to call This Week in What Has Science Done For Me Lately? All right. This comes in from Dan Linder. He says, hello, TWIS team. Here's my What Has Science Done For Me Lately entries. Of course, there were 2 entries, and I just copy pasted the whole thing because I like them. And so I just put them in there. Aha, entry 1. Years ago, after learning of the TWIS podcast, I was fortunate enough to hear a suggestion to use a salt as an abrasive to rub burnt on food off of pots and pans. The next day, we had one such event, and sure enough, it worked well. We continue using it to this day on our cast iron pans. Using dish soap on cast iron is a big no-no, apparently, because it removes too much of the oil seasoning every time.
Who did that? That wasn't me.
The more you know. I probably said something like that. Cast iron, I love it. Entry 2, related to what has science done for me lately? There was a segment a few weeks ago talking about how much non-Homo sapiens had actually invented that we take for granted. You asked for ideas of things that were invented well after that point that us Homo sapiens could call ours. I propose that modern ice cream is that invention. It required the domestication of cattle for milk, cultivation of specific plants for sugar, as well as the free time to churn the freezing mixture for extended periods of time. Not to mention the ability to do this in non-freezing climates by harvesting ice and keeping that into the summer. I can't provide facts backing this up, but I'd like to think it was hours. Thanks.
You know, I really appreciate, and I think I agree with this. Ice cream is not a thing that's ever come up in the archaeological record of the ancient hominids.
Nobody
that
else.
Would be really but as soon as I say this, there's going to that means we've jinxed it. It's like turns out Denisovans were big. But
ice how would you tell the difference between ice cream and like just milk and sugar?
If you got the milk and sugar and you were in an icy climate, I think it would have been a natural.
Yeah.
Okay.
And you're walking with your bucket of milk and your cup of sugar, and you tripped and you spilled it on the snow and you made ice cream. I mean, that's not that.
No, I love it. I love it. I think that is the one thing that we can verify has been claimed now by current modern humans is ice cream. I like it.
Anybody who thinks they have something else, let us know. In our What Has Science Done For Me Lately section, you can send me an email, Kirsten@thisweekinscience.com, or leave me a message on our Facebook page. We want to keep filling this part of the show with your letters, with your songs, with your poems, your odes. Please keep sending them. We need new ones. Come on.
Infographic, we could narrate it to the podcast listener. Just try to describe it really well.
That might be more difficult.
Yeah, we can do it.
Right. We'll work on that. Okay. But this brings me to my first story of the evening. If you're narrating something to somebody listening, you're using words to paint a picture that they can visualize in their brains, right? So you're essentially putting a picture into their heads. Scientists have really tried to do this using optogenetics. Carl Disseroth and researchers in his lab and related to his lab have used the technique that he came up with, optogenetics, to stimulate neurons in the visual system of the mouse brain in a way that makes the mice behave as if they had actually seen something. So it's like putting a fake image in the mouse's visual cortex that makes the mouse go do something. All right, so let me tell you how they did this. It's really a fascinating study. So first, optogenetics is this technique where they modify neurons. They put a gene into the neuron that makes the neuron respond to laser light. Historically, this has been blue light. And so it's always been optogenetics. It's this blue light. Well, researchers, the Disseroth lab, they went searching for other molecules that could be involved. And they found a new light responsive protein called CHR-mine, cremine.
Green lights.
This protein is better than the other channel rhodopsin proteins that they had been using because it can be activated by much less light, and they're able to use a very simple red laser to stimulate neurons.
Pew, pew.
So it makes it it makes it more accurate. So there's not just like light dumping everywhere in the brain. They're able to pinpoint things more specifically. So they used they set up these mice with this new optogenetic technique, and in the meantime, they trained these mice in this situation where there was they were shown either horizontal lines or vertical lines. So there are specific neurons in your visual cortex, also the visual cortex of mice. Yes, yours. There are neurons whose job it is to respond only to vertically oriented stimuli, like vertical lines, and others that respond only to horizontal lines.
I think I got more.
So there are neurons that respond to very specific things. These are these orientation stimulated neurons. So they trained the mice when they see horizontal lines, lick a water spout. And other mice, if you see vertical lines, lick the water spout. And so they had these different mice licking the water spout based on what lines they saw. And then they made the lines dimmer and dimmer. And they kind of got to the end of the mouse ability to detect the lines visually. So the lines were so dim that they weren't licking the water spout anymore. At this point, they started stimulating the neurons using the optogenetics. So they had recorded from the brain to find out which neurons were the ones that were responding in the brain to either the horizontal lines or the vertical lines. And then they targeted those neurons specifically, and they started stimulating just small groups of them, like 10 or 20 neurons at a time. So this is really fine-tuned targeting of neurons. And they found that if they got rid of the visual stuff entirely, stopped actually showing a little picture in the real world of horizontal or vertical lines, but only used the optogenetic technique to trigger up to like 20 neurons, that was enough to make the mice go lick the water spout in the properly trained situation.
Right. So the properly trained situation. So it's 20 neurons, but it's feeding into a cascade of trained behaviors. Yes.
Yeah. And so that is the thing. So small population of neurons feeds into this much more complex behavior. And so what they're thinking is that there are these cascades that take place in the brain. And so more complex tasks could be studied possibly using very small assemblies of neurons, and that we could maybe study things like memory or look at other perceptions like smells, touches, and tastes to be able to really figure out how the neuronal systems work and which neurons are involved in creating behaviors. My dystopic sci-fi take on this whole thing is, hey, somebody's going to start implanting things in people's brains to make them hallucinate stuff.
Right. Or like it could be a hat that you put on where the holodeck is then a real thing.
Right.
You could opt in. But my question is, how do we know that what they're doing to the mouse brains is what is making them see a thing instead of making them do a thing?
Right. So that is the big question. We don't really know that. We know that they did the thing when they saw the thing. When we knew they were seeing vertical lines, they licked the water spout. We know that happened. And then, we stimulated the neurons that are supposed to be the vertical orientation neurons, and then the water spout licking happening. So it is correlation of behavior to activation. We don't actually know this because we can't ask a mouse if they are seeing this. But
so if we're going to go sci-fi,
but they're recording in the brain, everything they said they recorded in the brain seemed exactly the same as the visual input situation. Visual input.
If we extrapolate to this far off sci-fi potential, would it potentially be possible to rig up a synthetic brain or a real brain, but rig it up to a system which basically would project what the brain sees? Is that a thing in crazy far off sci-fi land?
Oh yeah, to basically make brain TV.
Yes, so that you you could have receptors. You could test it so that you could show them a thing and make sure that's what is stuck out on the projector, right? So you could make sure that system works. And then you could manipulate that brain and see what comes out the projector.
Yeah, so they have already done this with some animals. I think the experiment I remember from several years back used cats, and they hooked cat brains up to, or it might have been bunnies, cats or bunnies, but they hooked them up to a video screen, and it wasn't a high resolution image, but they were able to get a recording of what was in the visual cortex.
See, that's what I want. Then I'd believe this. If they could hook up the mouse brains and show some, even just a blob, it'd be like, oh yeah, I can believe that's vertical lines. Sure.
So what's odd is I have a different take on this. What I was hearing was that there's a big set of data out here, and you have this response on the other side, which is a big set of response activity. To connect it, you need a trigger that says, okay, I've seen this, now I do this. Right. And it sounds to me like what they've narrowed down to is triggering that response mechanism without the input.
But so they were specifically targeting neurons within the visual cortex. And so if you're going to think about the behavioral loop, the trigger of the behavior, the movement that the animal would have had to make would have gone through, there would have been another loop going away from the visual cortex, probably down into the amygdala or thalamic area, and then back up, probably the thalamic area, and then back up to the motor cortex. So they should have been, I think, so there's a couple of different branches to what's happening. You're right.
Right, but what it sounds like, okay,
they weren't just stimulating the motor cortex.
No,
they weren't just stimulating.
They're doing something else. It sounds to me like they have targeted. They've narrowed down to that region that says, okay, if this visual input is this, then there's a reaction you should have. Mm-hmm. But that reaction trigger, sort of, I keep saying the word trigger. I don't know if there's a better way of saying it. The response should be this if you get this input. But without having that input, they've triggered what the response should be by targeting a specific region, which means unlike the ability to visually see it, you wouldn't need to visually see it. You're sort of circumventing the actual first experience, and you're going directly to the response trigger of having seen an experience.
So this is the interesting question to me, though, because vision does not occur in our eyes.
Right.
Perception of vision occurs in our brain. Our visual cortex allows us to put together all of those bits of light that come into our retina and up our optic nerve. And they're just random bits of data about light and color and orientation and, frequency. And it gets turned into the picture. There's our visual cortex is like the digital encoder, right? It's got the algorithm also in it that allows the image. And so the image itself should be in the brain at the highest level of the visual cortex. The percept should be there.
Mm-hmm. And if it's not physically there, the brain can create it. Anybody who's ever had a dream with imagery and interaction can
a hallucination. It's like a hallucination if you visualized anything, close your eyes and visualize, right?
Yeah.
It's
still there.
It's still there. And so the brain does not know the difference between its own activity and activity that was stimulated by something else. It's just activity. And so if
my brain can tell the difference. The difference. But
that's the idea. That's why hallucinations can be so frightening, is that reality, your brain is producing the perception of something that's not really there. And your brain doesn't know. You have to come around to figuring out that it's a hallucination. But in the meantime, your brain does not know the difference.
Oh.
Activity in the brain is just activity in the brain. That's one of the coolest things to me, is that, There's stimulation that comes to us through our proprioceptive and sensory system. There's stimulation that happens just within the brain. And part of this research actually could help us find out more about hallucinations and how these little tiny populations of neurons may be getting triggered by some random blip of activation. You know, like an itch on your nose accidentally triggers some random population of neurons in your visual cortex, and suddenly you see a blip of light out of your left visual field. That it's that kind of thing happening that shouldn't be linked. Maybe there are explanations for how these little tiny populations of neurons can have much larger perceptive effects to us. Yeah. Beyond that, parenting brain. Parenting brain is old brain. It goes way back. According to a new study that is out this last week in the Proceedings of the Royal Society B, researchers looked at frogs to find out a little bit more about the brain and parenting. Not all amphibians parent their children. However, some poison dart frogs really take care of the eggs, clean them off, carry their babies on their back. They caregive. And so we want to know, or researchers wanted to know, what is the difference? Where in the brain does parenting come from? What makes a species or an individual give care or parent? So the researchers took a whole bunch of these little frogs while they were carrying their babies around in the water, and then immediately took their brains because they needed fresh brains that had just been stimulated by parenting. They froze the brains, sliced them up, and then discovered that an area of the brain called the preoptic area lit up in caregiving frogs, but not those who don't give care. And this preoptic area is also something that's important to parenting in vertebrates and us mammals. So the fact that we share this area being active during parenting with frogs suggests that this is an area of the brain that evolved in some way for this caregiving behavior a long, long time ago. Another area of the brain, the medial pallium, it's
okay, I'm unfamiliar with this. Can you point to the general
medial pallium? But
right there in the middle.
An amphibian brain is set up much differently. Medial off to the side. It's in the middle medial side. But the amphibian brain is arranged much differently from the human brain.
Yeah, like this.
It's like squished. The amphibian brain is squished flat, and our brain is like layered up on top of itself. We're rolled in a ball like a burrito. So the medial pallium is similar to the hippocampus, which is for memory and memory consolidation. And that was also important in the brains of the frogs with tadpoles. And so maybe it's because they have to move their tadpoles around. We don't really know why, but that was another reason. And there were some differences between male and female caregiving frogs. So they found some neurons. There's a particular neuron that's tied to actions for grooming pups in mice. And when they looked at it, they only found activity in this particular neuron in one species of 3 species of frogs that they looked at. So there is some variation in the system, and not all of the species are the same. And there's probably, as in so many things we've looked at, a lot of
convergent evolution.
Thank you, convergent evolution going on in the system.
So I think this is really good. This is a really good pointer to the idea that the basics of parental care are not convergent, right? So when you think about it, you have these different types of animals evolving into new space, into new niches. And the amphibians were the first ones that walked out onto land, had this huge amount of space, all this food in the form of plants and also invertebrates like bugs and stuff. And so as they're exploding into this space, out in the ocean, broadcast spawning, pretty successful, just kind of floats its way out there. When you're on land, you have to be very careful about where you put your gametes. Yeah, a little more accuracy needed. So then you have to put this entire idea of where do I put my stuff, right? Which is starting to show up in fish, which I will talk about in the animal corner in a little bit. So the idea of where am I going to spawn starts to show up in fish. But once you're on land, you really have to be specific. And then from there, you really have to be specific about where you want to have your babies. Are there predators here? Is there a food source? So even if you're going to leave them and never take care of them again, there still has to be some careful decision making involved in where I'm going to put my babies. And then you go on, and as you go further through evolutionary history, you see more and more parental care because there's less and less babies, and there's excuse me, there's advantages to taking care of your babies. So you can kind of see, if you look through evolutionary past, you can see a very clear logical progression of parental care. And I think it's very cool that you can find out exactly in the brain where it's hanging out in these frogs.
Yep. And if you don't happen to have these particular areas of the brain wired for parenting, you're probably not going to be a parent. At least not a parent who cares. That's for sure. Hey, Justin.
Uh-oh.
Are you hanging out near your locker?
Oh, yeah. So, okay. This is, so CRISPR is the thing that everybody knows what CRISPR is. It's to some extent, it's table salt. What is it? A table salt phrase? It's the thing that everybody knows is a thing in the world. Coming next, LOCKR, LOCKR, which is short for latching orthogonal cage / key protein, with the R emphasized in protein to get LOCKR. All right. Well, okay. It's a bit of a stretch there, but it is a completely artificial protein switch that can work inside of a living cell, which can commandeer the cell's internal circuitry, can control, modify gene expression. What this means is you can have a cell implanted in your body under this that can, scientists show that LOCKR can be programmed to modify gene expression, redirect cellular traffic, degrade specific proteins, control protein binding interactions. Researchers also use LOCKR to build new biological circuits that behave like autonomous sensors. These circuits detect cues from cells' internal, external environment and respond by making changes within the cell. So this is sort of they're saying akin to the way a thermostat senses ambient temperature and operates your central heat or cooling system to shut off or turn on to achieve a desired temperature. These sorts of things have been designed before. Synthetic genetic circuitry is a thing that's been being worked on. But what this does is a little bit different than just circuitry. It's creating a protein that's going to have a set sort of scheme of activities, and you can put it into a living cell and determine whether or not it activates, which is very different. There's a quotey voice from El Samad, who's a professor of biophysics at University of California, San Francisco, co-senior author of the study. The ability to control cells with the designer proteins ushers in a new era of biology. In the same way that integrated circuits enabled the explosion of the computer chip industry, these versatile and dynamic biological switches could soon unlock precise control over the behavior of living cells and ultimately our health. Health is, of course, where they are laser focused on this. They're looking at improving the CAR T technology so that you can not only modify cells, but give them a very deterministic activity once they're activated. There is no counterpart in the natural world. So CRISPR is a thing that we discovered by discovering something that a virus bacterial interaction within the genome. This is completely synthetic biology. These proteins were made from scratch with a purpose of an activity that they would do. This is basically the first of its kind ever conceived on the planet. So they had a version of this that they installed in yeast, and they were able to show that genetically engineered fungus could be made to degrade a specific cellular protein at a time that of the researchers choosing. They could turn it on and say, okay, at this time point, we want you to degrade this protein. They then redesigned the switch and also demonstrated the same effect in lab-grown human cells. So this has a couple of layers to go still. But one of the things that is in the video, not in this report here, that they point to specifically is things like tumor targeting in cancer patients. Being able to degrade things that the tumors need for their sustenance and replication, you can put into place and allow it to get to the place where you want it to be activated and then turn it on remotely. This is not anything that has been possible before in the history of biomanipulation, I suppose. So fantastic. And it's the University of Washington is who came up with this. And there's sister papers that are out of UCSF that have found additional applications for the basic technology. But LOCKR, we're going to be hearing a lot about this in the probably next 3 to 5 years of TWIS.
Absolutely. This is, it does, like you mentioned, CRISPR. It's good. It's going to be one of those technologies that will be made use of by other researchers. They're going to be like, what protein can I stick to it? What am I going to latch it to? How am I going to modulate whatever system? And people are going to start playing with it.
It's a platform. So you can design a protein that will have whatever interaction to its environment you like. And you can
that can be triggered at some point.
You can hide it within another protein, which is the cage element. And you can decide when to activate it, which is really fascinating.
Yep. That is cool. Do you know what time? It's time to activate something that we like on this show.
What do we call it?
Blair's Animal Corner.
It's Blair!
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
Got Blair.
I have an amazing story about anemone fish, also known as clownfish. Justin, what do you know about clownfish?
I only know the movie.
Yeah, well, what did you learn in the movie? You learned that they live in an anemone.
Anemone.
And if that film was accurate, when Nemo's mother died, Marlin would have turned into a female. Yes, they are sequential hermaphrodites.
What?
They start out male. All clownfish start out as male. And the biggest, baddest clownfish in a specific area transitions into female and becomes the dominant female in a monogamous pair for life. So each pair makes their home in an anemone, which protects them from predators with its stinging tentacles. They appear, as far as we know, to be immune to their stings. Females lay eggs, males fertilize them, just like we were talking about. After that, the mother defends the nest. She protects it from predators, and the father tends to the egg. Sometimes there are other non-reproductive male fish allowed to live on or near that same anemone with the mating pair. If for some reason the female disappears, her male mate begins almost immediately to take on female behaviors, such as aggressively defending the nest, and the next largest male next door can move in to become her mate. So,
I don't know why it is I love that the biggest, baddest, most macho guy on the block becomes the matriarch.
Yes, that is correct.
Of the community.
Yes. So this is a study out of University of Illinois. There's psychology professor Justin Rhodes is a behavioral neuroscientist who wanted to see where this transition first begins. Now, if you were to guess, either of you, at what point would you consider the male anemone fish to officially be female? What would it be?
When he couldn't find a mate.
So you're saying the second the female dies?
Yeah, like as soon as there's nobody to mate with, and you're telling me it's an enemy sausage party. I'm like,
yeah.
Okay.
So I'm saying, at what point do you classify that individual as female?
When reproductive aspects change.
So that would make sense, wouldn't it?
Right?
But this specific study was looking at what changes first and what starts the cascade to turning female. Is it the gonads? Is it the brain? Is it behavior? Is it physiology? So the way that they figured this out is they set up experiments in the laboratory where they paired male anemone fish together and tracked their development. They followed 17 pairs of anemone fish. Within minutes to hours of being put together, one of the two males emerged as dominant. So basically they fought and fought and fought until somebody won. And whoever won began to start behaving as female almost instantly. So their original hypothesis was that as soon as it was kind of decided, okay, this is going to be I'm going to be the female. I'm going to be the female. Then immediately their gonads would start changing. But that's not what they saw. They looked at brain structure that regulates gonad function. This is the preoptic area. It's much bigger in females than in males in anemone fish. It is roughly twice the number of neurons. They track the changes in that area and the changes in the gonads and in their blood hormones. They counted neurons in the preoptic area, took blood samples to look at their sex hormones, and examined the eggs to look at the proportion of eggs to testicular tissue in their gonads. Within 6 months of being paired with a male, the dominant male grew their preoptic area to the size that is indistinguishable from females. So in 6 months, this part of their brain was completely female. After 6 months, this part of the brain is completely changed from a male brain to a female brain, but the gonads still did not change.
No.
So they're still male gonads. But what Rhodes says, that I think is so funny, is if you ask other fish, they'll tell you that's a female. So their behavior and everything about them outwardly tells you, yes, this is a female. So this is especially interesting because only 3 of the dominant fish in the 17 pairs actually transitioned all the way to reproductive females within the course of the study with viable eggs in their gonads. And only 1 of the 3 laid eggs. So the rest of them appear to be in a holding pattern where the female brains were fully functional, but they still had male gonads. They followed the fish for 3 years. And so the majority of them still hadn't finished their transition. They don't know, quote unquote, what they're waiting for. It could just take that long, or they could be waiting to grow bigger so they can reproduce more eggs because they put boys together. So they were both smaller. So it's possible they're just waiting to grow up big and strong so they can defend their anemone and lay lots of eggs. Or they say, Maybe they don't have the right chemistry as a couple. Maybe if they were out in the ocean, they would have ditched each other by now. We don't know. But what's really interesting here is that it is a very clear demonstration that brain sex and gonad sex can be uncoupled.
Yeah, so
absolutely.
I immediately thought of human anthropomorphizing, whatever.
Anthropomorphizing, yeah.
Thank you.
But what we do know is that there are sex hormones produced in the brain. There are sex hormones produced in the gonads. And if you remove the gonads, or if you remove, say, the uterus in the case of a hysterectomy for women, then there are still, it doesn't take away all the hormones in the body. There are still sex hormones, because brain sex and the gonadal sex, they can be separated. And but this is a very interesting natural model system to look at this in, to be able to see the effects on the brain and really tease apart how things change and why they do.
And I think to Justin's point, just the human side of this thing, which is not the point of this research
at all.
Right? But the thing that is interesting here is, if you know that in nature, brain sex and gonadal sex can be decoupled.
Yeah.
That is true. That can be true in other species. I'm not saying for sure we found it, but it can be true in other species, which can also explain certain situations that we have run into as humans, where people identify as one sex, but were born another physically. So there is something interesting there. I don't know the science behind that. I don't know if the science has been done to show anything is similar between this and that. But I just think it's a very interesting parallel and could be an interesting avenue of research.
And also interesting because it's an archetypal life form. Like, there was fish before there was most anything. There were fish before there was anything on land. So you're talking about a life form that this study was conducted in, that is, and essentially it's as evolved as anything else that it currently exists. But it's also.
But fish came first.
Fish came first. So anything that you see in fish, you should be able to see in other life forms all the way after.
Well, unless they lost it. Yeah.
Unless
but yes.
With gills.
But, yeah.
Okay. So not gills and everything.
Yeah.
Yeah. So now we've talked about what comes before the babies. I want to move on to parenting. Kiki was just talking about parenting earlier, in particular, parenting in birds, and more particular, yellow-legged gulls. So this is a study from Universidad de Vigo in Spain, looking at gull embryos' response to parental warning calls. So we see a bird on a nest, they see a predator, they call out a warning call. Are they calling to other birds, or are they calling to the eggs they're sitting on? Recent research builds on prior research, which showed that embryonic birds, amphibians, reptiles, insects can receive sensory information in the egg, in ovo, in utero, whatever it may be. So they can definitely receive sensory information while they are developing. This research wanted to look at warning calls on the eggs and how this would affect their development, and extra wrinkle, how what one egg hears might affect another egg in their same nest. I will explain. They collected 90 gull eggs from nests along the shores of Salvora Island and brought them back to the lab for testing. They separated eggs into individual 3-egg clutches and incubated them. So in each of these 3-egg clutches, they pulled out 2 of them to expose them 4 times a day to a test, either recorded adult warning sounds or silence. Still a test. So then they took those 2 and put them back in with the first egg untouched. They reported that the embryos exposed to the shrill warning calls would vibrate when the recordings were played and continue vibrating for some time, even after they were returned to their incubator. They suspected that the vibrations could be felt by the nestmate that had not heard the recording, so that this vibration would pass on information that they got when they were removed to their nestmate that didn't hear it. To find out, they monitored the embryos after they hatched. The birds exposed to warning sounds took longer to hatch. Maybe they were being extra safe coming out. When they finally did, they were quieter than the chicks that had been exposed to silence. They crouched lower when they were exposed to perceived threats. And they were smaller overall and had shorter legs. So all advantageous changes to a space riddled with predators. The clutch mates of chicks exposed to the recordings, so the ones that weren't exposed but were next to them, had all the same differences, even though they never heard the warning calls themselves. So this means that most likely they felt the wiggling that the embryos were doing of, oh God, did you hear that, Hawk? Of nearby embryos, and responded as if they had heard it themselves.
That's crazy.
Yeah.
What?
It's crazy, but also it's super not to me. It makes sense. If they're developed enough to have anything like a brain in there, why wouldn't you respond to stimuli and that would release fear hormones that would change their development, right?
Yeah. These are birds we're talking about, but people put their headphones and little speakers on their pregnant bellies so that their children can have a particular audio and experience in the womb of music, or you talk to the child in the womb because those vibrational sounds can make their way to the developing child, and those vibrations that can affect things. So it's really interesting to see this in a species in the wild and having long-term developmental influences.
So maybe don't watch a slasher film while you're pregnant. I don't know.
I mean, who knows? The baby wouldn't know what that is. A baby wouldn't know what that is.
Screaming? If there's just a lot of screaming, they might know.
Yeah. I mean,
just a thought.
I wonder for us, the gulls, what the chicks and the eggs are responding to the sounds of predators. Mm-hmm. So these are birds that are predated, and it's triggering a response pattern that will protect them, possibly, make them harder to catch, make them hide faster, will protect them as they're young birds and as they're developing. Yeah. Humans don't have lots of predators, do we?
Yeah, if we just played like lion roaring sounds.
Yeah, I'm just kind of wondering what sounds would be the ones that would influence, your chance at death, right? What would do that?
Yeah, I don't know. Well, anyway, for the birds, which is actually what this is about, adaptation to perceived threats while still in the egg. Pretty cool.
Yeah, that is very cool. Yeah. I love it. Staying in the egg. We are towards the end of the show. Got some quick stories here. Hey, an apple a day can give you lots of bacteria.
Oh, no.
Even if you wash it.
Yay. Yay.
Yeah, good bacteria or bad bacteria?
Good bacteria. It's fine.
It's just bacteria.
Fine.
Lots of bacteria. So researchers decided to look at the bacteria on and in apples, and they've published in Frontiers in Microbiology. Previously, research has looked at the fungal species that are on and in apples, but nobody had ever looked at bacteria before. So the researchers were like, let's check this out. People eat raw apples. They don't cook them. And so apples, like other fruits, end up potentially populating your intestines, your gut with bacteria, because if it's not cooked, the bacteria is not killed. So you've got bacterial transfer. So they checked it out and they looked at the stem, peel, flesh, seeds, and the calyx, which is that little flowery bit at the bottom, or it's where the flower used to be. And they discovered that regular sized apple, not with one of the big giant ones, but a regularly sized apple has about 100 million bacteria. But if you discard the core, you'll only end up taking in about 10 million bacteria.
So do I want to eat the core or I don't? I'm not.
Wait,
the majority of the bacteria are in the seeds.
Is that a quantitative or is that a diversity of?
Oh, good question.
I believe this is a quantitative.
Yeah, sounds like it.
This is quantitative, not diversity. And there were pathogenic bacteria found in most of the conventional apple samples, but none from organic apples. But beneficial lactobacteria that we like in our probiotics, it was the opposite. There was lots of that in organic apples and none in the conventional apples. And there's other methylobacterium. The researcher says, known to enhance the biosynthesis of strawberry flavor compounds, was more abundant in organic apples, especially on the peel and flesh, which in general had a more diverse microbiota than seeds, stems, or calyx. So even though there are quantitatively more bacteria in the seeds of the apples, the diversity is greater where it interfaces with the environment more often. Yeah. Anyway, microbiome, pretty big on that apple and might be a part of the reason that an apple a day keeps the doctor away. Maybe. We'll see about that. Anyone else have a story? Who's next?
So following studies from Florida International University's College of Business, and was published in the Journal of Marketing, which on the surface sounds like just the sort of thing your parents would have warned you might encounter someday, with instructions to turn around and walk briskly in the other direction. And they were right to say so. However, as much as it slightly pains me to say so, psychological sciences directed at consumer triggers is likely one of the most sophisticated and if not in every case completely misdirected for somewhat nefarious purposes, benefits of psychology, or most astute psychological professions. So in this, they looked at the effects of temperature on humans facing a decision. And so the study analyzed housing auction data, which kind of sounds like bidding on who gets bragging rights or profiting on the suffering of others at the hands of a commoditized housing market, but I slightly digress. The data measured the impact of temperatures between 67 and 77 degrees, which they called moderate, and temperatures as high as 82 degrees, which they put into the high temperature category. So they also had two sort of categories of these transactions. One was an auction setting and one was a negotiation. So one is you're bidding against others to see who gets to win the thing. And in that, those in the high temperature category offered more money than those in a separate auction environment who had a lower temperature. They also found that in a negotiation setting, higher temperatures led to harder negotiators. They wanted even lower prices. They were underbidding what would be the underbid at that point. And what they kind of came away with was higher temperatures lead to more aggressive activity. So if you're bidding in an auction environment, the aggressive thing is to try to get it by having a higher bid. If you're in a negotiation situation, the aggressive activity is to offer less for the negotiated, for the agreeable price. I found this really interesting because, of course, their advice is if you're holding an auction versus a negotiation, you might want to raise the temperature in the room to get the most from your client because business people are scum. And they don't, they could just have easily have said, if you are trying to be the client, you should opt for a colder room versus a warmer room.
Right.
You're right. They could have easily, but no, immediately their conclusion is if you're the one who's hoping to profit from, fine, that's who they are. I get it. But what I found really fascinating about this is also as simple as it is a temperature variance in human behavior. There's a correlation throughout history of wars being started in higher temperatures.
In hotter temperatures, yes.
And it's usually blamed on scarcity of resources, over water, over stuff like that.
No. That's what's actually wild about it. It is the assumption of resources that has also been attributed to this. So you've gotten to the spring, everything's planted and it's now growing. You go to war in the summer so that you can get home, hopefully, by harvest. Because the resources are being produced, you can also feed an army. That's one portion of this. But the other is the leaders of countries, whoever they are, the decision makers, make more aggressive decisions when it gets hotter. So these marketing people found this in a microcosm of a auction versus negotiation with completely different outcomes. But here's my takeaway from this. Here's where we can make this
my takeaway is that, hey, climate change is making it hotter everywhere. And in about a week, the Arctic's going to be as hot as Morocco. So awesome.
Okay. So here's how you, the listening audience, can actually make use of this. If you and your spouse, your loved ones, you and your children, you and your roommate, whoever it is, need to come together to make some decisions, crank up the air conditioning. Crank up the air conditioning. You will make less aggressive demands of the other. You will find more mediating positions to agree on something. If it's somebody you don't want to agree with, if you're going to the car dealership and you want to put yourself in the mind frame, put heating pads all over your body.
Right. Do it on a hot summer day.
You can actually self-manipulate yourself into being in a situation where people are more accommodating or less accommodating with this information.
Yeah. Cool. Who's next?
So do you know what else happens when it gets hotter? Animals shrink.
What?
Yeah. So we've talked a couple times on the show about different theories of how body size is shrinking in different populations due to climate change. One story in particular that has stuck with me that I actually teach in a lot of my climate change lessons now to kids is about mountain goats in Europe, where as they're pushed higher up in elevation, there's less and less free oxygen and the babies aren't growing as large or as quickly. So the entire population is actually shrinking in body size. But this study from University of Cape Town looked at a 23-year period between 1976 and 1999 and found that climate change is indeed shrinking the body sizes of mountain wagtails, which is a type of bird. And they think that this is because larger animals in general tolerate cold conditions better than smaller animals. So you would expect warming climate to be more advantageous for smaller animals. But there are other potential impacts, of course, because that makes them potentially more likely to become predated upon. It's an important indicator of fertility, so it could cause problems with sexual selection. It can mess with lifespan. It reduces lifespan sometimes. It reduces its ability to survive in stress and food shortages or drought. And so these are all things that potentially could impact species that are shrinking because of climate change in a very short amount of time, and other factors of their survivability is not adapting at the same rate. So it is something that in this particular species of bird was measured. It has been hypothesized in a lot of different species, but I just thought I would throw that out there since it was a pretty large scale study with a very clear answer.
Birds are shrinking. It's getting hotter.
Are shrinking from climate change.
What's interesting, there's been a question as to whether airplanes are going to have issues in the changing atmosphere because the density of the atmosphere will be changing as things heat up. So planes may have to lighten up to be able to stay up.
No. Oh, no.
It might affect airplanes as well. But that brings us to the end of another episode of This Week in Science. I want to say thanks again to our guest tonight, Dr. Sarah Parcak, whose book is Archaeology from Space: How the Future Shapes Our Past. Fun read. And I would like to also thank everyone who's involved in making This Week in Science happen. Thank you to Fada for helping with show notes and monitoring our chat room and social media. Also to Gord McLeod for helping in our TWIS chat room, and Identity4 for recording the show. Thank you also to our Patreon sponsors. Thank you, Paul Disney, Richard Onimus, Ed Dyer, Ed Andy Groh, Stu Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, Mark Mazzaros, Jack Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Eric Knapp, Richard Brian Condren, Dave Neighbor, Gene Tellier, John Gridley, David Williams, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney, Tiffany Boyd, John Bertram, Mountain Sloth, Seth O'Gradney, Stefan Albaron, John Ratnaswamy, Dave Friedel, Daryl Myshak, Andrew Swanson, Paul Ronovich, Sue Doster, Dev Wilkinson, Ben Bignell, Richard Porter, Noodles, Kevin Reardon, Christoph Zuknarek, Ashish Pant, Ulysses Adkins, Artyom Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Slazewski, Jim Drapeau, Greg Riley, Sean Lamb, Ben Rothig, Steve Leisman, Kurt Larson, Rita Garcia, Marjorie, Gary S., Robert Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and EO. Thank you for your support on Patreon. If any of you are interested in supporting us, you can find information at twis.org. There's a link to Patreon there. You can go directly to patreon.com/thisweekinscience. You can also subscribe and tell your friends to subscribe. We have the links at twis.org. And again, next week, we will be broadcasting online live at 8 p.m. Pacific time on twis.org/live. You can watch and join our chat room, but don't worry if you can't make it. You can find all of our past episodes at our YouTube channel or twis.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or you can look for This Week in Science in anything Apple Marketplacey.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twis.org. That includes information on our guest if you're interested. And while you're there, you can also make comments and start conversations with the hosts and other listeners.
Or you can just contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line. Otherwise, what will happen, Blair?
I believe your email will be spam filtered into oblivion.
Into oblivion! You can also hit us up on the Twitter where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that came to you tonight, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science.
science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And this week in science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This week in science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 This week in 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 science.